All right, let's jump right into this explainer. We're going to uncover the tiny, invisible heroes keeping us alive every single second. We rarely stop to think about the incredible, bustling, microscopic world inside our own bodies. But right now, as you're taking this all in, millions of tiny biological machines are working tirelessly just to sustain you. Today, we're zooming way in to look at these machines, known as enzymes, and decode the really strict rules they have to play by.
To really appreciate what these machines do, think about this for a second. Imagine sitting down for a totally normal lunch, say a sandwich. Now, what if you had to wait almost an entire month for your body to process that one meal? No way, right? You literally wouldn't survive. Your body needs energy and nutrients way faster than that.
Well, that's the stark reality. Without our biological helpers, digesting that single sandwich would take a grueling 2 to 3 weeks. But with them, that exact same process is wrapped up in about 4 hours. That is literally the difference between life and death. Our metabolic reactions absolutely have to be fast enough to keep us alive.
So what are these helpers? They're enzymes. We call them biological catalysts. They're biological because they're made naturally inside our living cells, and they're catalysts because they absolutely turbocharge the rate of chemical reactions. And honestly, the wildest part? They do all this heavy lifting without ever getting changed or used up themselves. They just keep going.
Okay, so here's our roadmap for today. First up, the lock and key. Second, testing the temperature. Third, the pH puzzle. And finally, the danger of denaturation.
Let's start with Section 1, the lock and key, enzyme specificity.
Now, enzymes are incredibly picky. Because they're proteins, they fold into very specific 3D shapes. The molecule that an enzyme actually works on, we call that a substrate. Notice how enzyme 1 has a shape that perfectly matches that yellow substrate? They're complementary. But enzyme 2, the green one, it just won't fit. It's a fundamental rule of biology. Enzymes are highly specific to one particular substrate.
Think of it like a microscopic assembly line. The enzyme has this special docking station called the active site. The substrate slots in perfectly, literally just like a key sliding into a specific lock. When they connect, they form what we call an enzyme-substrate complex, and that is the exact moment the magic happens. The substrate gets rapidly broken down or joined together to form brand new products.
To break down this whole factory process, it's basically 3 steps. First, just bouncing around randomly, the substrate collides with the enzyme and fits perfectly into the active site. Second, boom, the chemical reaction is instantly catalyzed. And third, the new products are kicked out. Since the enzyme itself doesn't change at all, its active site is immediately empty and ready to grab the next substrate. It's just this endlessly repeating cycle of efficiency.
Moving on to Section 2, testing the temperature, effects of heat.
Let's play lab detective for a minute. To see how heat affects enzymes, we can test amylase. That's an enzyme that breaks down starch. We mix amylase and starch at different temperatures, and we use iodine to check our work. See, iodine turns blue-black if starch is present. We just check the mixture every single minute until the color stops changing, which tells us the enzyme has totally finished breaking down all the starch.
And the data here is fascinating. Look at this difference. At a cool 20 degrees Celsius, it takes 9 minutes for the enzyme to finish the job. But warm it up to 40 degrees, and the enzyme speeds way up, finishing in just 3 minutes. But, and this is crucial, look what happens when we crank the heat up to 60 degrees. 0 digestion. None. The reaction halts completely, the iodine just stays blue-black forever. The amylase has flat out stopped working.
Which brings us to Section 3, the pH puzzle, acidity versus alkalinity.
So heat is a big deal, but enzymes are also super sensitive to pH. In a much faster-paced experiment, we use buffer solutions to strictly control the pH level of our amylase and starch mix. Checking every 10 seconds this time, we're basically hunting for the optimum pH, the absolute sweet spot for this enzyme to do its thing. Turns out, enzymes are just as picky about pH as they are about temperature. A neutral pH of 7 is the clear winner here, taking only 60 seconds for the reaction to complete. Make it slightly acidic at pH 5, and it slows down to 80 seconds. Slightly alkaline at pH 9, 90 seconds. But check out that extreme alkalinity at pH 14. Just like with extreme heat, there is absolutely no reaction. The enzyme is completely defeated. Okay, let's look at why that happens in Section 4, the danger of denaturation, breaking the bonds.
What's actually going on here is a process called denaturation. Remember how we said an enzyme's 3D shape is held together by chemical bonds? Well, when you push an enzyme past its optimum conditions, like way beyond our body's perfect 37 degrees Celsius, the intense heat or extreme pH literally rips those bonds apart. The whole protein structure just collapses and deforms. The lock is essentially melted. Because the active site is permanently warped, the substrate can never fit in there again. The machine is broken beyond repair.
And that leaves us with a really profound thought. Every single metabolic reaction keeping you alive right now relies on these delicate, incredibly specific locks and keys. And they are fragile, too hot, or a major pH shift, and they denature for good. So if our microscopic machines break that easily under the wrong conditions, just think about the immense, complex effort your body is putting in right this very second to maintain that perfect 37-degree environment. It's a fascinating, precarious balancing act happening inside us every single moment. Also, we can digest